First, I would like to thank the organizers, especially Dr. Anuradha, for this wonderful opportunity. And also, I thank other speakers of the day, and the previous session was really interesting and intriguing because our talk is going to be more on the genomics, but more in the basic science perspective. And I also concur with Dr. Amit, who had mentioned that we need to learn a lot. Even in the basic science perspective, we need to learn a lot about how these sequences have been generated, what are the real biologies and what are the artifacts in order to understand both the tumor biology as well as trying to predict good drivers. So in this talk, I will try to summarize few points that we could do with the genomics data set. So ours is a majorly computational lab, so maybe my biology will be a bit limited, but I will try to explain as much I understood. So the data, genomics data that is currently available are mostly generated from the tumor at the very late stage or the tumor that don't respond to the chemotherapy. When we do sequencing from these tumors, we not only see the mutations that are important for the tumor development, but also see other mutation that has been accumulated over the lifetime of the individual, because these are all somatic tissues. And this is the problem that may be alluded as information overloaded, partly because we have plenty of mutations to deal with, and we need a specific way to figure out which one could be a likely driver. And here we are trying to come up with some computational approach to do that. And at the same time, given that there is enormous information available about the mutation, can we learn some basic biology from that too? So the way we are doing is, we take the genomes that are available from the large consortia like TCGA, STC, and then some specific studies from the national group. And we try to understand how the mutations patterns are in these tumors so that we could learn what is more expected and how they are distributed in the genome. And then we use that information to create a background model, which is what is my expected baseline mutation rate in a given genome of a tumor type. Then I could try to see whether the mutation I see in a particular individual is surprising or not surprising. And then we can try to design whether it could be a likely driver. Okay, so starting off with the mutation patterns, there has been some seminal studies from Alexandrov group where they have shown that across tumors, there are specific mutation type more prevalent, and they call it as the mutational signatures, and each of them could be associated to a specific mutational process. For example, we see a lot of C2D mutation at the CpG sites, which could be associated to the spontaneous deamination at the CpG sites. Or in case of lung, we often see C2A mutations, which is associated to the benzoyl and which is affecting the guanine residue, and it leads to more C2A mutations. So this is interesting to know what mutations are expected in a particular tumor type and its more prevalence, and also we can know from the sequence context where do they occur more. So this could tell us that from this figure, I can say that the mutations at CCC context where the base C to A gets mutated is more common than expected. So given this information, one can predict where I could see a mutation in the genome. But the challenge is our genome is not just a linear sequence, it is being bound by some proteins. Okay, so it's been packed. So there are some regions which are heavily packed with the proteins, and there are some regions which are less packed. So because of this differential packing, the DNA is also differentially exposed to the DNA damage and the repair. So by looking at the mutation distribution, people have found that the open chromatin region, which are less bound by proteins, tend to have a lesser mutation as compared to the tightly packed region because the accessibility of the DNA to the repair mechanism. And in a recent study, we also found that even in the open chromatin regions, there are some regions which are constantly bound by proteins like transcription factor, which is needed to regulate the gene regulation, that also induce the mutation rate variability in the local scale. So taking these things together, on one side, we know which mutations are more frequent in a particular tumor type. And then on the other side, we know how they are distributed in the genome, which is shaped by the chromatin context. So our next speaker, Dimple, one of my colleagues, she will talk about more on this chromatin aspect. So together, what we see here is how chromatin shapes the distribution of the mutations in the genome. So if we take those information and we can predict a baseline mutation rate, we can assign a probability for how likely a particular base is going to be mutated. So in the panel below, you can see that I have highlighted two or 4 different sequence contexts, of which 2 of them are the same. So for example, the green one TCA, and there is another one TCA. So you could see the probability to observe mutation at one site will be much lower, whereas the same context, but just being present in a different genomic context, have a higher probability. So this kind of probability could tell us that whether if you see a mutation in a particular region, is it more surprising or not? Because the sequence context and the chromatin is influencing the probability to see the mutation. How this is useful? Once you have this information, now we can go and check whether the mutation we see at these sites are present across all tissues that can be explained by the VAF, and whether it is present in more than one individual. That could tell us the recurrence rate. So if you see a mutation that is highly recurrent and have a high VAF, but present at a site which is very unlikely to get mutated, then we can say that the mutation is rare and there is a selection acted on top of that to enrich that clone to be overgrown. So this is the way we try to define the driver mutations. So I'll just give you an example with some genomic analysis that we have done from the whole genome study conducted by Pan-Cancer Analysis of Whole Genome, PEACOCK Consortium. So they have gathered around 2,500 whole genomes across 37 tumor types. This is the large-scale whole genome available at the time, and we were trying to predict drivers at different sites by taking the information of how likely a particular site is going to be mutated, and then the selection analysis. So in the interest of time, I will just focus on 2 tumor types, which is the lung squamous and lung adeno. And although the sample numbers are very low because of the whole genome data, we were able to predict the mutations that has already been reported from the previous TCGA exome studies. The additional thing is that given this is the whole genome, we were able to identify all possible drivers that could happen, like point mutations, amplifications, deletions, fusions, and cis activating genomic rearrangements. So that is the advantage of the whole genome, where we could comprehensively identify all possible driver identification. So we missed some of the driver events here, like for example, EGFR. It has been found only in the 3 samples, and because of this frequency, it is not listed here. And the possible reason could be is the coverage or the number of samples that have the mutations in it. Otherwise, in the lung squamous carcinoma, the mutations that is expected, like P53, CDKN2A, are more prevalent, which is 42 out of 47 samples. And whereas in lung adeno, we see very less driver events. And if you look at the type of alteration that is seen in each gene, you can see the variability across them. For example, there are some genes where P53, as expected, it has the mutations followed by a copy number alterations. CDKN2A, it's mostly deletion event, and SOX2, which is often amplified. So in the previous lung cancer studies, many of these events has been defined separately because the technique that used are possible to identify these events separately. But with the whole genome, we could find all of these events from a complete analysis, from one comprehensive analysis. So moving on, given that we have defined drivers in each sample, now we can ask how many patients had a driver mutation and what is the average driver mutation in these samples. And what we noticed is that at the pan-cancer level, the number of driver events that we found was around 4, which means that each individual have at least 4 to 5 main driver events. And this we could find in 91% of the sample, which means virtually all tumor had a driver event. The remaining 10%, which we could not explain due to the low coverage, or it could be driven by non-genetic mechanism. And in most of these tumors, we also see a whole genome duplication, which is an event in addition to these driver alterations. We see the whole genome gets amplified. So some of these drivers will also get amplified through this process. But we don't know whether this whole genome duplication itself is a separate driver event. But when we go to the lung squamous carcinoma, we find that many of the tumors have a high number of driver events. So in each individual, we can see around 8 main driver events, and almost all of them had one of those driver events. And these tumors have a high whole genome duplication rate as well. Whereas in the lung adenocarcinoma, we see around 5 driver events, and we were able to identify that in 97% of the samples. and roughly half of them have the whole genome duplication as well. So collectively, the whole genome sequencing has a lot of information to infer, both in terms of point mutations and beyond that. The challenge is it is quite expensive, and sometimes the coverage may not be as uniform or in a high depth in a targeted region. The additional message is that we haven't found any recurrent driver mutation beyond the coding region. Although this is a whole genome sequencing, we haven't found any driver event in the non-coding region. The only gene that we have found is the TERT, which had a promoter mutation. But in a majority of the sample, the TERT is amplified. Only in a couple of them, we see the promoter mutation, which has been initially found in melanoma, but now has been found in other tumor types as well. So the non-coding drivers are sparse, but this may be related to the limitation in the way that we find drivers in the non-coding regions. So once we define the drivers, together with the copy number information, can we try to time which mutation has occurred in the early stage or the late stage of the tumor? This is to understand the biology and the clonal evolution. And this is one particular sample of the lung squamous cell carcinoma. And here, by looking at the copy number together with the mutation, we could say that the CDKN2A is the early driver event, and then followed by the different driver events that has happened, like T53, KM2C, not C1. And these were defined based on how many cells within the tumor tissue shares these mutations, and then whether the copy number has happened together with these mutations or not. So collectively, what at the computational way, what we could do is, from the sequencing data, we could learn the mutation patterns, how they are distributed in the genome, and we can try to find the drivers, and then we put them in the context of which driver event has occurred during the lifetime of the tumor evolution. Although this kind of studies has been done in the whole exome and the whole genome, where the data quality and the coverage is good, there is still a lot more to be done if you go to a different tissue samples. For example, FFPE, where we have a lot of artifacts that could come from the processing, which has been discussed in the previous session. So in our group, what we are trying to do is we are trying to incorporate those error models as part of the background calculation and try to predict what is the likely driver, given there is a lot of noise in the background. Hopefully, once we have this method, we can run through the newly sequenced samples to look for the driver mutation. We are still beyond creating the NGS report, but I think there is a lot of research need to be done at the basic level to understand this different biology behind the genomics and the distribution of the mutation, and to develop some effective methods to list out the driver event. With this, I would like to acknowledge the lab members, funding agency, all the consortia which has generated this data and made it publicly available, and thanks to you for your patience. Happy to take questions later.